This presentation is going to, obviously from the title here, focus on the ovulation to the end of week 2. It goes through some different stages, and we have focused already our efforts on, we know a lot about ovulation, so this will be a little recap slash moving forward. So here's a basic diagram of the female reproductive system with several things identified. You have the vaginal canal, which we will discuss in previous videos. We'll talk about the concept of the fertilization and going through that. From here, right where the cursor is, it would be the cervix, and we are going to identify the left and right ovary here with the different parts of the fallopian tube. Obviously, ovulation is going to take place. We're going to highlight the oocyte that would have been ovulated from this ovary. So we're talking day 14. Keeping days straight is kind of important for there's a lot of things happening during this process, and we're going to assume that there is fertilization that would eventually take place. So following the fact that this would have been kind of the graphene follicle, if we use that term from the previous idea, so the graphene follicle would have been ruptured on day 14. That graphene follicle would eventually have given rise to the corpus luteum, and that corpus luteum is what is producing that progesterone that is going to be the hormone that is responsible for this endometrium lining, the uterine lining, to become more vascular, meaning it has a lot of blood supply for the eventual implantation of the embedding of the embryo down the road. So let's zoom in on that structure. You can kind of see here, there's the magnifying glass. Day 14 happened. We've got the different portions. In a few slides, we'll see the different portions of the fallopian tube, but we will zoom in and see what the contents of this structure. So here's that zoomed in image. It has some of the important structures that we've talked about already, the corona radiata. The corona radiata is composed of these purple granulosa cells. And those granulosa cells were at one point part of the follicle, that graphene follicle, where there were some tissues, and this cellular grouping here would be the tissue of the graphene follicle interior. It was at one point called the cumulus ovorus, remember that term from the ovary. But here you have the corona radiata, which is all of the granulosa cells, plus this orange component back here, a very, very important protein structure called the zona pellucida. And so the big gray structure would be the egg cell, and this would be your nucleus of the egg. All right. So that's what all the components are that would have been ovulated and accepted by the fallopian tube. Here's a real image of what that would look like. So there's a lot more granulosa cells than the previous image has shown. This is what would be the tricky part for getting all the way through the fallopian tube. If it had all this stuff on it, it would get stuck as the diameter of the fallopian tube is not wide enough for this entire structure to fit through. But through the process of fertilization, we are going to see what happens if there actually were to be fertilized. If this did not get fertilized, a few days down the road, there would be a degeneration of all this cellular debris. So it would just probably collapse based on the fact that it wasn't a thriving, living structure that would kind of keep that form. So zooming in even more, you could notice that there in faint surrounding is that zona pellucida with a very distinct egg cell in the middle here. But that faint area right around, you can kind of see the zona pellucida. All right, the structure now, the egg has made its way into the fallopian tube. These little projections, recall, are called the fimbriae, and the fimbriae are what guide the egg from the ovary into this duct, the fallopian tube, where fertilization would eventually take place. So the fimbriae are not labeled, but the fimbriae are the little tiny projections, little fingers. And now we have, of these four, I talk about these three most important. The area that is the widest, that the egg has already gone through in this image, is called the infundibulum. So we can say that that is the widest portion of the fallopian tube. The ampulla is a very important region. It's kind of smack dab in the middle. It's narrowing, as you can see here. But what will occur, the ampulla is really as far as that entire structure can make it with the corona radiata. This is where fertilization will eventually take place. So the sperm, which would have entered from this direction, making its way through the cervix and bypassing and getting through, because it is a much smaller cell, it can narrow and get through this narrow region very easily. But the egg cell is much harder and hardier of a cell, so it's going to be more difficult. So it stops here at the ampulla. The isthmus is that narrow region. If you know the term isthmus, it in geography it's a narrow bridge between two continents or two land masses. So it kind of fits here the narrowness of the isthmus. So here's that picture that was kind of insinuating would occur. We have the sperm that are entering through the vaginal canal, and we have a more detailed fertilization video that if you need to think about that, but this is just the generic idea for right now. So what we have here is a zoomed in image, obviously, of the sperm and some regions that are very, very important. We have the tail, which is a microtubule. It's a 9+2 arrangement, and those are doubled microtubules all in a concentric ring. The mid or the head region. The head region contains the nucleus, which is a haploid nucleus. So in a typical human, there would be 23 chromosomes in there. What is not really highlighted is the midpiece. The midpiece, this area where the cursor is located, would be very concentrated in mitochondria due to the fact that in order for this tail to propel, a lot of energy is required. So there would be a high concentration of mitochondria there. In front of the nucleus, we have a yellow acrosomal cap, or the acrosome. This contains digestive enzymes, which play a huge role in the process of fertilization. So these digestive enzymes will eventually give rise to the enzymes that bury itself through that corona radiata and get through the zona pellucida. So it's very, very important that you have this region. On the outside, in very faint pink, they call it gunk. It's probably layers of glycoproteins, which we don't get into specific names of them, but there are these attachment pieces that cover the sperm during its voyage in the uterine lining. And eventually, the process that we're going to highlight in the next picture is a very important process called capacitation. So the picture shows that the gunk, the pink stuff, is kind of breaking up. And that's the idea of capacitation. And capacitation is twofold. It is the cleaning of the head of the sperm, which is clearly identified here. And this will also help with the mobility of the sperm. The speed at which it moves changes after capacitation. So that is kind of the two important parts. And this would happen naturally within the fallopian tube. But that is a very important process for it to be able to fertilize. If you don't capacitate, you don't have the ability to penetrate the egg. So unfortunately, there's a lifespan that happens. After capacitation, the sperm has a really limited lifespan. So if the sperm enters well before the egg is ovulated and capacitation takes place, the sperm will die well before the egg has the potential. So it's all about this timing game for the sperm to enter at the right time. The sperm now has exposed a region where there is going to be a receptor for the zona pellucida. So the zona pellucida itself has receptors for the sperm. They are called the ZP3 receptors, which in other videos we discuss in great detail. But the zona pellucida has receptors. So the sperm also has to kind of have receptors for it to connect as well to the egg itself. So there's an image of the zona pellucida that is connected to the sperm. So there will be a connection piece between two very important receptors. I focus more on the receptor on the zona pellucida, which I just mentioned would be the ZP3 receptor. So that's where the sperm would be located right now. 3 just stands for there's multiple receptors. We focus on the third one primarily in this course. But the sperm now has attached to the zona pellucida. This would cause what is now the second part of fertilization, which would be the acrosome reaction. So the acrosome this is the part where the acrosome would bust open and those digestive enzymes would be released. Another sidebar This is, again, details that fall in the fertilization video, but this would be the moment where calcium ions would just flood into the sperm. And that is going to also cause this reaction of the acrosome. So it all kind of is related to some ion diffusion that goes into the sperm to signal, hey, this process has started. So let's get moving. And here we have it jumped forward in this kind of general video of the process of fertilization and implantation. But what starts to happen, the sperm is going to latch onto the zona pellucida through a series of reactions. There's actually a process which doesn't get highlighted in this video, but it's the process of preventing polyspermy. And polyspermy would be the entrance of multiple sperm into the same egg. We don't want that to happen. So they have skipped in time to show that what occurs, there's a influx of the nucleus only. So this blue area, the nucleus of the sperm has now entered into the egg. And what this does is it causes a reaction. This is kind of the final step of fertilization, where inside of the cell, the signal has been reached by the egg to say, hey, there is a nucleus that has entered from a sperm. So what occurs technically is the smooth ER inside of this egg is going to also release some calcium ions. And those calcium ions are important, and the smooth ER is not identified here, but the smooth ER will release the calcium ions, and those calcium ions go and trigger two different events. The first event here are the cortical granules. They're labeled. They're the pink. Technically, they are just lysosomes that have a very specific role. So lysosomes, you remember from an intro to bio course, contain also digestive enzymes. What happens at this moment, those lysosomes are triggered by the calcium to go to the outside of the egg. So they're all concentrated out here. And what they'll do is they will fuse with the membrane. And with that fusion, all of the contents of the lysosome, or the cortical granule as it is called, are released to the outside. If you can picture exocytosis. So this is all kind of background biology. But the lysosome will fuse with this membrane, and the contents will then go on the outside of the membrane. And those enzymes are going to start to release these granulosa cells. And that's called the cortical reaction. The second thing that calcium ions did was they triggered to this nucleus to, hey, finish meiosis. Because at this point, since fertilization hasn't occurred, if you knew anything about female meiosis, it doesn't complete until fertilization takes place. So the calcium ions from the smooth ER trigger this to completely finalize meiosis. So that's a lot going on in that slide. There is the cortical reaction. With those zona pellucida is still there intact, but the cortical granules, their responsibility is to get rid of all of this extra debris. And that poses an important reason due to the fact that now the size of the overall structure has really diminished. We were discussing the size can't get through the isthmus, but now with all of those granulosa cells that have been released, they could fit through the isthmus. So there it is. It can start the journey down the fallopian tube to fully enter your uterine cavity. Simultaneously, sorry, there's a 7-minute bell, but simultaneously, since fertilization has taken place, as it is going down the fallopian tube, yes, it's traveling, but inside there's things happening. As soon as it got fertilized, we call that a zygote. But now we're going to start going through mitosis very, very quickly, cell division. We're going to make identical nuclei. So these were originally one zygote with one nucleus, but now we have two cells, each with their own nucleus. And we just go through. So there we have in a real image, zona pellucida still present. 4 cells, 4 cells, 16 cells. So this would be called a morula now. And all this is happening within the same confines of the zona pellucida. So what's not happening? The cells are not going through any growth. So they skip the G phases of interphase. They go right through mitosis after mitosis because they're trying to increase the size of or number of their cells. They can't increase the size of their cells due to the fact that the zona pellucida is kind of keeping it into this volume. So we're ending on this slide because I'm at a 15-minute little increment, but we'll continue to talk about some of the details of the future.